Hello students, in the previous class we studied about the transverse section of dicot root and monocot root, and we studied about the differences between the dicot root and monocot root. Now in this class I am going to explain about the transverse section of dicot stem. Right. So when we take the transverse section of a dicot stem and observe it under microscope, we can see this type of structure. Under microscope, so which is called ground plan. This is called ground plan of dicot stem. Ground plan of the T.S of transverse section of dicot stem. Right. So in its transverse section, when we want to observe each and every tissue very clearly, each and every tissue present in dicot stem very clearly, we observe one part of this dicot stem. In this ground plan, we observe one part of this dicot stem present in ground plan. So this part or this part or this part. So that one part is this structure. This structure is representing one part of the ground plan, having one vascular bundle, like that. One vascular bundle along with peripheral tissues. Right? Okay. So in the transverse section of dicot stem, it consists of three layers, three regions. They are epidermis, cortex and stele. In the transverse section of dicot stem, three regions are present. They are epidermis, cortex and stele. Now let's start with epidermis. So epidermis is the outermost covering of plant organs. So here also in stem also it is the outermost layer which is made up of epidermal cells, which are thin-walled parenchymatous cells, and these are compactly packed without any intercellular spaces. And this epidermis is covered by a cuticle, a waxy layer called cuticle. So this cuticle is formed by cutin. Cuticle is formed by cutin, which is secreted by epidermal cells. So what is the function of this cuticle? Function of this cuticle is to reduce the rate of transpiration which occurs through the surface of young stems. Right? So the epidermis present in stem is cutinized epidermis or cuticularized epidermis. Right? Okay, some of the epidermal cells present in this epidermis show extra growths, not outgrowths. In case of root hairs, we call them as outgrowths. But in case of stem hairs, we call them as extra growths, which are growing above the epidermal cells. And these extra growths are called as trichomes, which are also called as stem hairs. And these stem hairs or trichomes are multicellular, and the function of these trichomes is to reduce the rate of transpiration, and they may be secretory also. So two functions, one is reducing the rate of transpiration and they may be secretory also. Right? So this is about epidermis. Now this epidermis is followed by cortex. This cortex is present between epidermis and stele, or the region of the stem which is present between epidermis and pericycle is called as cortex. And again this cortex is divided into three subzones. They are hypodermis, general cortex and endodermis. Now let's study one by one. So hypodermis. Hypodermis is the outermost region of the cortex which is present just below the epidermis. And this hypodermis is few layered, that is 3 to 5 layered, which is made of thick-walled collenchymatous cells. That means hypodermis of dicot stem is made of collenchymatous tissue. So the walls are thicker towards their corners, and hence there is no intercellular spaces in hypodermis. And the cell walls consist of thickenings which are made of cellulose, hemicellulose. Cell wall is made of hemicellulose and pectin. So the pectin which is present in the cell walls, in the collenchymatous cell walls of this hypodermis, gives shiny appearance, and hence they appear golden yellow in color. They appear golden and yellow in color when we observe this hypodermis under microscope. Right? This is about hypodermis. And what is the function of this hypodermis? Hypodermis is involved in providing mechanical support. Function of epidermis is to provide protection. Function of hypodermis is to provide mechanical support to the young stems. Right? How it provides mechanical support? It prevents the breaking of stems due to high wind velocity. Right? Okay, this is about hypodermis. Hypodermis is followed by general cortex. Now, this general cortex is few layered, which is made up of round or oval shaped parenchymatous cells, and these parenchymatous cells are loosely packed, and hence intercellular spaces are present in general cortex. In epidermis, there are no intercellular spaces. In hypodermis, there are no intercellular spaces. In general cortex, intercellular spaces are present due to the loosely packed parenchymatous cells, and these cells are thin walled. So general cortex is made of parenchyma. This general cortex is multi-layered, which is followed by endodermis. Endodermis is the innermost layer of cortex. Innermost layer of cortex, which is present between general cortex and pericycle. Right? This endodermis is single layered, which is made up of thin walled parenchymatous cells again, and now these parenchymatous cells of endodermis are involved in storage of food in the form of starch. Hence this endodermis is also called as starch sheath. It is also called as starch sheath. Whereas in case of root, whether it is dicot or monocot root, endodermis consists of Casparian strips. But here in the endodermis of stem, dicot stem, these Casparian strips are absent, but it is involved in the storage of food materials in the form of starch. Hence it is called as starch sheath, right? Okay, now this endodermis is followed by stele. Stele includes what? Pericycle, vascular tissues, that is phloem and xylem, and then pith, right? Okay, so here let's study pericycle first. So pericycle is present below the endodermis, and this pericycle, I am writing the details of pericycle here. This pericycle can be homogeneous. It is either homogeneous or heterogeneous. Homogeneous pericycle is either homogeneous or heterogeneous. Homogeneous or heterogeneous. Okay. So what do you mean by homogeneous? Homogeneous means if the entire pericycle, this one. So epidermis, hypodermis, cortex, pink colored one, endodermis, followed by pericycle. So this is pericycle, which is present above the vascular bundles, right? Okay. So if the entire pericycle is made of only one type of tissue, that is called homogeneous pericycle. If the pericycle is made of two types of tissues, that is called heterogeneous pericycle, right? If it is made up of only one type of tissue, that is sclerenchymatous. If it is homogeneous, it is made of sclerenchymatous tissue. That is called homogeneous pericycle. If it is heterogeneous, some part of this pericycle is made of parenchyma and some part of this pericycle is made of sclerenchyma, right? That is called heterogeneous pericycle. Okay, let's observe why it is heterogeneous, how it is heterogeneous. Okay, these are the vascular bundles, right? These are vascular bundles which are arranged in the form of ring, right? So in these vascular bundles, this is phloem, this is xylem. Phloem and xylem are separated by cambium. In between phloem and xylem, cambium is present, right? Above this vascular bundle, pericycle is present. That means above phloem, pericycle is present. So, the part of the pericycle which is present above the phloem or above the vascular bundles is sclerenchymatous. I am talking about heterogeneous pericycle. In case of heterogeneous pericycle, it is made of two types of tissues, sclerenchymatous and parenchymatous. The part of the pericycle which is present above vascular bundles, that is above phloem, is sclerenchymatous, and the part of the pericycle which is present beside that is in between vascular bundles, so this part is sclerenchymatous and this part is parenchymatous. Again sclerenchymatous, which is present above the vascular bundle, that is above the phloem, and this is parenchymatous, again sclerenchymatous, again parenchymatous. So this sclerenchymatous pericycle which is present above the vascular bundle, above the phloem of the vascular bundle, is forming a cap-like structure. Hence it is called bundle cap, which provides mechanical support because it is made of sclerenchyma. Function of sclerenchyma is to provide mechanical support, right? So pericycle can be either homogeneous or heterogeneous. If it is homogeneous, the entire pericycle is made of only one type of tissue, that is sclerenchymatous tissue, right? If it is heterogeneous pericycle, it is made up of two types of tissues, that is Sclerenchymatous and parenchymatous. The sclerenchymatous pericycle is present. The part of the pericycle which is present above the vascular bundles is sclerenchymatous. In between is parenchymatous, and again above sclerenchymatous, parenchymatous, sclerenchymatous, parenchymatous. So the sclerenchymatous pericycle which is present above the vascular bundle is forming bundle cap, a cap-like structure above the vascular bundle, which is called bundle cap. So this is forming bundle cap. Bundle cap, right? So its function is to provide mechanical support. Next, and one more point here, and this pericycle is in the form of semilunar patches, like this patch, one patch. So this is one vascular bundle, like this. This is one vascular bundle. Above this vascular bundle, pericycle is present. In this vascular bundle, actually what is present? Phloem, xylem. In between phloem and xylem, what is present? Vascular cambium, and above this phloem, pericycle is present, which is sclerenchymatous pericycle, which is in the form of semilunar patch. That means half moon. And after this vascular bundle, one more vascular bundle will be like this, and in between pericycle will be there, na? That is made of parenchymatous tissue, right? So this is about pericycle. Next, coming to vascular bundles. So in Dicot stem, these vascular bundles are arranged in the form of ring. So arrangement of vascular bundles in the form of ring is the characteristic feature of Dicot stem. And this arrangement is called as U-stele. So arrangement of vascular bundles in the form of ring is called as U-stele, right? So the type of stele present in Dicot stem is U-stele. Why it is called U-stele? U-stele because vascular bundles are arranged in the form of ring. Hence it is called as, the type of stele is called as U-stele. Right. Now let's study the details of vascular tissues, vascular bundles. Okay, I am writing it as VB, vascular bundles. So the vascular bundles present in Dicot stem are conjoined, open type of vascular bundles, which are with end-arc type of xylem. I'll explain why conjoined, open and end-arc, right? So conjoined, they are conjoined because this is one vascular bundle, right? This is one vascular bundle. So they are conjoined because both phloem and xylem are present together in the same vascular bundle, forming one vascular bundle. So this is phloem, this is xylem, forming one vascular bundle. This is phloem, this is xylem, forming one vascular bundle. Hence it is called as conjoined, where both the phloem and xylem patches are joined to form single vascular bundle is called as conjoint vascular bundle. And it is called open vascular bundle. Open vascular bundle because in between phloem and xylem of this vascular bundle, cambium is present. So this is the cambium which is present between. What is this cambium? Cambium is meristematic tissue, right? So this meristematic tissue undergoes cell division during secondary growth of plant, during secondary growth of stem, which results in increase in the girth of stem, right? So conjoint because both the phloem and xylem are joined. Name itself suggests conjoined. They are joined to form the same vascular bundle. Open because between phloem and xylem cambium is present, which results in secondary growth, right? Okay, and end-arc because the type of xylem is end-arc xylem because protoxylem elements are, protoxylem is towards the center and metaxylem is towards periphery. Protoxylem is towards center and metaxylem is towards periphery. Protoxylary elements consist of small vessels, small sized vessels with narrow lumens, protoxylem, and metaxylem large sized elements with broad lumens. So protoxylem is towards center, metaxylem is towards periphery, right? And hence it is called as end-arc, where protoxylem is towards center, end-arc type of xylem, right? So this is about the vascular bundles present in Dicot stem. Okay, so in between these vascular bundles, so these are vascular bundles. This is one vascular bundle, like this vascular bundles are present. In between these vascular bundles, parenchymatous tissue is present. Parenchymatous tissue is present in the form of rays. Like this rays will be formed, okay? So in the form of rays, these are called medullary rays. right? So these are called medullary rays. So these are medullary rays. Are you getting my point? These are medullary rays. Okay. What is the function of these medullary rays? Function of medullary rays is to transport water and minerals and even food materials towards radial side. So medullary rays are involved in radial conduction of water, minerals and even food materials, right? Okay. So this is about medullary rays. Next, at the center of the dicot stem, parenchymatous tissue is present, which is called pith. So the pith is made of parenchymatous tissue in which the cells are round shaped or oval shaped, which are loosely packed, right? And in case of dicot stem, this pith is well developed and it is conspicuous. Okay. In case of dicot root, pith is not well developed. It is small. In case of dicot root, pith is small. It is not well developed and it is inconspicuous, which cannot be seen clearly under the microscope, right? And sometimes that pith in dicot roots may be absent. But in case of dicot stem, pith is large. Pith is large. Centermost parenchymatous tissue, pith, which is large and it is well developed and it is conspicuous, which can be seen very clearly under the microscope. So this is all about dicot stem, right? Due to the presence of cambium in vascular bundles, this dicot stem shows secondary growth, due to which it increases in its girth. Okay, so this is all about the dicot stem, transverse section of dicot stem, and in the next video I will be explaining about monocot stem, transverse section of monocot stem. After studying the transverse section of monocot stem, let's make the differences between dicot stem and monocot stem. Hope you understood this concept. Thank you, students.